The role of the pathologist in the CDX kind of a device, we are not replacing pathologists whatsoever.
When a project starts as a manual score, it can fail because the human eye imposes artificial categories and biological gradients.
One of the key capabilities of computational pathology is also the ability to precisely assess the entire region of a protein expression intensity within a tissue section.
We are just empowering them with more quantitative, more precise and reproducible tools. So that will require a major cognitive shift.
What we see is that the more we refine our algorithms, the more we understand how to train these algorithms to quantify staining heterogeneity, providing detailed and objective measures of uniformity or non-uniformity of a biomarker distributed across a tissue.
The manual score is too noisy to find a correlation. So the biomarker looks non-predictive, but measuring protein density with the algorithm on a continuous scale eliminates the subjectivity, this variability, and noise present in human assessment.
Learn about the newest digital pathology trends in science and industry, meet the most interesting people in the niche, and gain insights relevant to your own projects. Here is where pathology meets computer science. You are listening to the Digital Pathology Podcast with your host, Dr. Alexandra Zhurov.
Welcome, everyone, and thank you for joining me for this webinar. I'm Dr. Alex Zhurov, founder of Digital Pathology Place, and today we're talking about something that is quietly changing how companion diagnostics work. And what that means for pathologists. For decades, companion diagnostics have rested on visual IHC scoring, and that model launched precision oncology, but it's now reaching its limits. To help us understand what's happening and where it's going, I'm joined by three experts from Roche. First, Dr. Gordana Juric-Sekhar, anatomic pathologist at Roche with deep involvement in digital pathology projects across the company. Gordana, welcome.
Thank you so much for the invitation to be here, Alex. And as you mentioned, I'm Gordana Juric-Sekhar, and I'm a physician, board certified in anatomic pathology and neuropathology. I began my career in academia before transitioning to industry 5 years ago when I joined Roche. I'm involved in various aspects of companion diagnostic development through multiple avenues, primarily the evolution of computational pathology and the development of new biomarker scoring algorithms.
Thank you so much. I'm also joined by Saleh Miri, PhD, the Director of Digital Pathology AI Algorithms at Roche, where he spent over 8 years leading algorithm development for histopathology. Saleh, welcome.
Thank you, Alex, for the invitation and having me on this webinar. As you mentioned, I'm leading the clinical digital pathology algorithm development at Roche for past 8 years, and very excited to be here talking about computational pathology as the next generation of companion diagnostics.
I am super excited. And we also have Purvi Gaglani, Regulatory Affairs Lead for Digital Pathology at Roche. And in the past 3 years, she was working closely with the FDA on submissions across computational pathology, companion diagnostics, and software as medical device. Purvi, welcome to the webinar.
Thank you, Alex. Excited to be here. Very, very interesting topics today, and being part of the team that has been able to interact with health agencies across the world when it comes to computational pathology, CDX, and software as a medical device. I think we're at the cradle of bringing something very new for our patients and everyone across the globe. So very excited. Thank you.
I am so excited about this topic because I've been following the development of this new way of doing companion diagnostics for, 2 years already. I don't remember when was the first time that it was publicly mentioned that this is happening. But the format today is a little bit different. We don't have presentation, but we've got 8 questions to work through over the next hour. And then we'll open up live Q&A. So let's get into it. I'm gonna start with the question to you, Gordana. My first question is, what does the companion diagnostic landscape look like today? And what data are we using and how it's being scored? Because that's going to set the stage for what's new.
Thank you. So despite the emergence of new technologies like next generation sequencing or let's say liquid biopsy, immunohistochemistry has been the foundation of companion diagnostics for decades, largely due to its practical advantages. While next generation sequencing processes the sample to identify mutations, immunohistochemistry allows a pathologist to review the sample composition and confirm protein expression either on the cellular compartment or on infiltrating immune cells. Also, immunohistochemistry is a standard procedure in nearly all pathology laboratories globally, and it's generally faster and cheaper than new technologies. I have to say that a clinical landscape has shifted from binary, positive, negative, to a spectrum of expression. Biomarkers are all visually scored. Pathologists currently use semi-quantitative scoring methods for companion diagnostic tests, which involve estimating ratios such as tumor proportion score and combined positive score, or assigning categories 1, 0, 1 plus, 2 plus, 3 plus, based on staining intensity. So the era of one-size-fits-all pathology has ended. The expertise of a pathologist is also pertinent, and training on specific companion diagnostic biomarkers is now a regulatory and a clinical imperative. For example, FDA labels for almost all new CDX assays explicitly state that the test must be interpreted by a qualified pathologist. Semi-quantitative scoring is the current gold standard for precision medicine, but it highlights a significant bottleneck. When a drug approval is tied directly to a specific assay, the pathologist's eyes become the ultimate gatekeeper for therapy. However, the challenge is that a single tissue section can contain, let's say, 50,000 or even 100,000 tumor cells. For a human is truly calculate, it takes hours per slide or whole slide image. Instead, we pathologists use visual bins or percentage ranges. Let's say, we'll say the tumor shows positivity between 40% and 50%. So, but also, we are generally better at identifying positive cells than accurately counting the total number of viable tumor cells, the denominator. However, if the denominator is wrong, the entire score is wrong. In the context of current CDX landscape, I would also emphasize the role of computational pathology, as its advancements have taken it beyond the experimental phase. So computational pathology can address some significant challenges, such as I call first point the borderline dilemma. In many clinical trials, the cutoff for a drug is set as a specific percentage, for instance, 75%. Humans cannot accurately differentiate it between 74 and 76% tumor cell expression in tens of thousands of tumor cells. However, digital tools can perform an exact cell count. The borderline patient receives a more accurate classification, reducing the risk of denying potentially beneficial treatment. The other point, which I want to also stress, is beyond protein expression. Most CDX assays only look at two things: how many cells are stained and how dark is the stain. This ignores the spatial context of the tumor. So computational pathology can measure spatial heterogeneity. It can answer, are the protein expressing tumor cells touching immune cells? Are they clustered together or scattered? Trials are already showing that these spatial metrics are often better predictors of drug response than simple positive-negative bins. So to summarize this overview, we are still talking about manual scoring, but the era of eyeball bin is coming to an end. The CDX landscape is evolving toward an integrated multimodal system shifting from, I would say, qualitative to quantitative science.
Thank you so much. So I want to highlight a couple of things that you said, because you started with estimating ratios. And well, like I have research experience with PDL1 scoring. And I remember like being trained on how to do the borderline cases. And I went to a specific training and they would show us these borderline cases. Doesn't matter how long we trained and how they, like how many hours they trained us. At the end of the day, half of the room was showing that it was below the cutoff and the other half, it was above the cutoff. So like, it's impossible. And the scores based on calculation, you said, but we never calculate, right? If we were calculating every single cell, it would take us pathologists hours per slide. There is no hour per slide, so you do these bins. And most of the time we're okay, but the borderline cases who are borderline samples from patients, and the decision on the borderline case is going to change, okay, what kind of treatment they're going to get. So that brings me to the second question that I have for you as well. Where does the current companion diagnostic model begin to reach its limits?
The current companion diagnostics model, which relies heavily on the manual interpretation of immunohistochemistry, it's approaching its limit because it essentially demands human vision to perform fundamentally mathematical operations. Yeah. So although this model initiated precision oncology, its current sensitivity is insufficient for next generation treatments, such as we are talking about, let's say, antibody drug conjugates. So the current model has critical limitations that are evident at certain points. Foremost is interobserver variability. Variability can be attributed to differences in a pathologist assessment, including tumor proportion score or combined positive score interpretation, staining intensity interpretation, and membranous versus cytoplasmic staining interpretation. For the tumor percentage positivity, for example, a cutoff is, let's say, 50%. And one pathologist would assess 45%, and another would call it 55 positive tumor cells. For the staining intensity interpretation, the cutoff is, let's say, 2 plus and 3 plus at 50%. And the distinction between 1 plus and 2 plus staining can lead to interobserver variability. Sometimes it's challenging. The third point, for example, for membranous versus cytoplasmic staining interpretation. For example, the cutoff is 50%. Membranous staining at any intensity. The assay is challenging to distinguish between membranous and cytoplasmic staining. This can lead to wide differences in interpretations. But I also would like to emphasize other points which may reveal limitations. Intraobserver variability. The same pathologist might score the same tumor differently, for example, today and tomorrow morning. Search fatigue. On large tumor resection, a small cluster of positive tumor cells could be easily overlooked, altering a score. Another point, which I also want to stress, is a low sensitivity assessment, the traditional staining intensity. We always talk about 0, 1 plus, 2 plus, 3 plus system has evolved. Now pathologists need to distinguish in HER2 and also evaluate ultra-low 0 plus, which includes any faint, barely perceptible membranous staining in tumor cells. So next, it's average positivity and heterogeneity. Tumor may reveal heterogeneous biomarker expression with areas of intense so-called hot and minimal or no so-called cold immunoreactivity. So a pathologist often averages these areas for a single score. For example, 15% average could mean 15% of cells are stained evenly throughout, or it could mean one small corner in 100% positive area while the rest is 0. So these patients respond differently to treatment, but the bin treats them equally at some point. And one point which also want to mention, it's failure to capture spatial context. A manual score provides a single number. For example, combined positive score is 30. But it doesn't specify where the protein is expressed. Two patients with the same 30% score can differ. Positive tumor cells might be clustered near immune cells in one patient, but isolated in the tumor core in the other patient. We now know that spatial proximity, a distance between T cell and tumor cell, predicts successful immunotherapy better than protein density alone. Manual scoring cannot measure these micron-level distances. I also want to add the decades-old staining intensity scale system, which is the gold standard for breast and gastric carcinoma, is an inconsistent predictive biomarker in advanced non-small cell lung carcinoma. In breast carcinoma, HER2 aberrations are typically associated with an overexpression of receptors on the cell surface. On the other hand, in non-small cell lung carcinoma, HER2 is generally characterized by a structural abnormality, and a genetic mutation causes the receptor to be permanently active regardless of receptor quantity. So a high 3+ score doesn't always mean that, for example, HER2 gene is amplified. And many patients in advanced non-small cell lung carcinoma with a IHC score of 0 or 1+ carry HER2 exon 20 mutations and respond to targeted drugs. It's interesting, yeah. And so I put many points together, but in summary, I can say that the current model reaches its limit at the threshold of human perception. To unlock the next level of precision, the diagnostic must shift from being our judgment call to a quantified measurement.
So that's like the clear visual limitation. And I also see the evolution of as we as a research community, I would say, as we discovered more biology, we tried to apply the pretty decent, robust method of visual assessment to something that's too granular for this assessment, right? And the first level is, okay, let's have us not guesstimate, let's have us have some computer-aided tools, right? To show us how much score is there, and then we can confirm. It's still the same of visual principle, but now computer is calculating this. And this has been there for HER2, for PD-L1, these computational companion, but that's different from computational companion diagnostic. I want to differentiate two things, because this is computer aid for pathologists. It basically does what this score was designed. It calculates instead of estimates. And this is the computer aided AI tool. And this question is going to be for you, Purvi. I would love you to explain to us how does a computational companion diagnostic, so truly computational, differ from this assistive or computer aided AI tool that pathologists are mostly familiar with, because a lot of these scores have a computer algorithm that can calculate, right? But we're going to be talking about something that is new and that differs. And I want to highlight this difference.
Thank you, Alex. Yes. What we've seen with the advent of computational pathology is the changing role that the pathologist has in the overall clinical workflow. And as you rightly put it in your question, the difference between what we're doing with computational pathology versus creating tools that have aided pathologists in making a diagnosis or an assessment is starkly different when it comes to the way the pathologist is involved and the amount of work they get to do when they're interacting with such a CP-based device. This particular device that has been the talk of the town, we see that the role of the pathologist is more focused on ensuring that the sample quality, the scan quality, the image quality is of a certain nature, and is allowing the pathologist to gain that confidence that whatever analysis comes out from a computational pathology workflow or an algorithm is something that they can trust. Whereas if this were a computer-aided scoring algorithm or something that was done to aid the pathologist in enhancing what they're doing, would probably be restricted to giving them an idea about, I think this is where the tumor cells lie. Now it's up to the pathologist to figure out whether they agree or disagree with that scoring, and also make amends to that. That is a very distinct identifier between what a computational pathology based algorithm for a CDX workflow does versus what it would do for a computer aided program or a tool that would just allow them to make their work a little bit easier. But what we also see is that with the advent of computational pathology that is facilitated by the tools that we have in digital image analysis has marked a transformative shift in the way IHC images have been viewed. There are better scanners now, there are better image management systems, and a lot more sophisticated algorithms that involve artificial intelligence, deep learning models that allow us to say that whatever scoring we're going to receive at the end of that workflow is a thoroughly vetted model that is giving you the confidence to accept the score that the algorithm is giving you. The challenges that Gordana described are exactly the challenges the computational pathology algorithms are trying to solve. This makes our solution more scalable. It allows us to get diagnosis to people around the world where pathology diagnoses have been challenging. They don't probably have the right kind of tools. They probably don't have enough amount of doctors who can help them. Having a decentralized workflow where you're sitting anywhere in the world, and as a doctor, as a pathologist, you can view an image from any patient anywhere, is where this marks the advent of what the future looks like for digital pathology. One of the key capabilities of computational pathology is also the ability to precisely assess the entire region of a protein expression intensity within a tissue section. What we see is that the more we refine our algorithms, the more we understand how to train these algorithms to quantify staining heterogeneity, providing detailed and objective measures of uniformity or non-uniformity of a biomarker distributed across a tissue. And it also allows to gain a level of resolution which has in the past been either challenging to the human eye in terms of assessment or in terms of quantifying it to a level that would become universal. And hence the challenges that Gordana mentioned, where you see a lot of path variability is the problem that this potentially takes away. When you couple softwares like this with AI and deep learning models, we see that it creates a very, very powerful diagnostic tool, and it permits the efficient, systematic investigation of any disease, any biomarker that has so far either eluded us or has been difficult to diagnose and hence quantify. We have also seen that with a new generation of drugs coming in, the need for precision medicine, the need for having very precise outcomes of your diagnostic test, of your companion diagnostic test, CP allows us to define those and gain insights into these new biomarkers and allow us to have companion diagnostic devices that can work on new drugs. So this indeed is that full solution where you not just get to see we are creating a tool that allows pathologists to do their work a little more efficiently, but actually taking away the burden of having to go through those 10, 15,000 cells, allowing to see them, the membrane intensities, the stain intensities, that is quite challenging and burdensome in terms of a workflow. At the same time, allowing them to then come up with a diagnosis that is very direct, precise, and has high sensitivity to a drug that we know would work for the patient. So, all in all, when we compare a CP based diagnostic algorithm versus a computer aided tool for the pathologist, there is a clear strength in developing tools like this and developing algorithms like this that truly allow the pathologist to scale their work and make it more sensitive and precise as we move towards new drugs and biomarkers.
Thank you so much. So the role of the pathologist in this landscape changes, and because they need to kind of collaborate with the new way of diagnosing, right?
Right.
It's going to be, okay, we have a precise tool, but is this tool measuring in the right place? Was this material even diagnostic? Did we do the correct sample? And basically, it becomes collaborative. One thing that you said, basically both of you, Gordana and you, Purvi, said, Gordana, you said IHC is everywhere, right? And we just mentioned, and computational pathology doesn't have to be everywhere to help those patients that would have IHC done on their samples. So I just want to have that in the back of our head, because there, and we'll have questions and answers for everybody, but basically we can help patients somewhere else than where their IHC was done. And there's this theme, okay, biology is getting more complex and we're discovering more biology, and we're going to be getting to what particular test we're talking about. But before we get there, Saleh, I have a question for you. Why does personalized oncology now require higher resolution biomarker measurement? What is there? What are we discovering, right? We just said at the beginning that actually this visual scoring did launch precision oncology, but I'm super happy that we're advancing. And why these higher resolution biomarker measurement requirement right now?
Yeah, thanks for the question, Alex. I think with what Gordana and Purvi mentioned, they set the stage nicely for us to relate to this question: why do we need higher resolution biomarker measurement? And I want to just highlight a few points from my perspective. I guess the modern targeted therapies nowadays, they are highly sensitive to subtle shift and respond to much finer biological gradient of the expression. And because of that, we do need precise tools like advanced technologies that can essentially, capture these minor fluctuations that ultimately they predict the therapeutic benefits and the patient response. So besides that, Gordana talked about this quite nicely, this continuous versus categorical change that we have. Mm-hmm. So the tumor biology itself is inherently continuous and heterogeneous, right? But the current companion diagnostics, this visual assessment and estimations that we have, it reduces this crucial data into simplistic categorical bins like 0, 1 plus, 2 plus, 3 plus. And this categorical scoring frequently fails to consistently predict therapeutic benefit, especially when we deal with more sophisticated and complex cases such as advanced NSCLC. One more additional point that I wanted to highlight also here is and Gordana touched on it, the limitations of this visual scoring. I guess achieving a truly reproducible and cell level quantitative signal requires moving beyond this visual estimation, because this visual estimation and categorical scoring inherently introduces high observer variability, as you also mentioned from your experience in PDL1 scoring. So no matter how much you train, no matter how much you read cases, there is still some inherent variability between and within pathologists. So that reader variability is always there, and computational methods are necessary to standardize the measurements and deliver the precision that is required nowadays for these modern therapies.
Mm-hmm. Thank you so much. So the biology that we discovered then translates into the therapies that are super sensitive, right? That's why we have a companion diagnostics. That's why we want to check, okay, who is this drug going to be good for, and who is not going to react in the desired way to the drug, right? And the cutoffs are like very granular, how to say, and you have granularity, like we said, is beyond perceptions, and that brings us to the thing that you said, continuous versus categorical. Categorical doesn't cut it anymore. We need continuous, and you mentioned also, okay, at the cellular level. So we are now, and that takes us to the marker that is kind of the hero of this webinar. That's the marker where we have this new companion diagnostic, TROP2, in advanced non-small cell lung cancer. This is going to be our example. And what is a computational pathology-based companion diagnostics for this particular marker? We kind of hinted to it, but I want you to explain what it does. And when you explain that, it's going to be clear why it's visually impossible to replicate it, because that is the thing that's happening now. Before, we were kind of decent in estimating, and here we reached a point where the granularity is so that we cannot estimate anymore. And we have to have this collaboration with the computer, with the computational tool. So let's talk about the TROP2 scoring. How does it look right now?
Sure. I can start from my side, and I will pass it on to Purvi to also talk about the regulatory and FDA perspective on this. So a computational companion diagnostics is a new class of regulated diagnostics where the final score is derived from, as I mentioned, algorithmic cell-level quantitative analysis. And this quantitative analysis, completely replaces subjective visual estimation. Rather than dividing the results into discrete categories like 0, 1 plus, 2 plus, 3 plus, this will be a truly continuous measurements of the protein expression. For TROP2 in NSCLC specifically, this involves calculating the normalized membrane ratio, or NMR in short. This is the metric that precisely quantifies the amount of TROP2 protein at the cell membrane relative to the total expression within the entire cell. And as you can see, this is completely different from the current standard, the visual estimation that Gordana explained quite nicely. So this goes beyond the current standard and goes to the individual cell level quantitative analysis and measure this ratio, which quantifies the membrane staining intensity versus the entire cell, which includes membrane and cytoplasm together.
Mm-hmm.
So I would like to also invite Purvi to talk about, from a regulatory standpoint, how is the entire system defined?
Yes, thank you,
Yes,
Saleh.
this is super interesting because that's like a new frontier of, how does it look from the regulators' perspective? What do regulators do with such new discoveries? And how did you interact with them as well?
The
If you can say
interactions, I
anything
can
about that.
say, yeah, the interactions have been very, very interesting. Let's start with that. We've
Mm-hmm.
learned a lot in our discussions, in our communications with the agency, not just here in the US, but across the world. So, all health authorities, one common theme that we see when we interact with health authorities across the world is everybody wants better solutions. Everybody wants precise solutions. And health authorities are willing to engage with the manufacturer to ensure whatever help they can provide, whatever guidance they can provide. If a guidance is non existent or if the device is as unique as ours, they're there to support you, they're there to help you. So it's been an extremely enriching experience, especially working very closely with the FDA on this particular device, gaining the trust and understanding how they review the device, knowing it's a very novel concept. It's a very novel technology. Having the experience to work with them on getting the BDD, which was the breakthrough device designation that is granted to medical devices that are meeting an unmet medical need with a unique technology, is what gave us the validation and the impetus to move forward in a direction where we know we're helping patients receive diagnosis that probably has been missing so far. One of the most interesting things that we learned is, if I compare the device that we're talking about right now to a traditional IHC based device that is visually reviewed under a microscope, we would probably be talking to FDA about an IHC kit for the assay.
Mm-hmm.
FDA would review the reagents, FDA would review the objective evidence associated with how the slides are stained, how the V&V was performed, and all of this would be by a human being looking at a microscope. That idea, that approach is completely changing here. Staining workflow or getting your slide ready has become a part or a component or an input to this new diagnostic device, where up until now, whatever they had been reviewing is now a system within a system of systems. So the perspective has largely changed, and what otherwise used to be an assay kit is now just a starting point for what the diagnostic workflow would look like. What we have seen is that one of the strongest recurring theme across our discussions with the health agency was ensuring that we do not work outside device definition. And for FDA
Mm-hmm.
especially, this was a device definition that started at us staining a slide all the way through the digital pathology workflow, generating a PDF with your biomarker results. And so you had a staining workflow, and then you had a digital pathology workflow where you're taking your glass slide, scanning it for creating a whole slide image for your image management platform, and then applying the algorithm to that image to come up with a biomarker score. Of course, there is the pathologist in the loop for the digital pathology workflow as well. And that is where the pathologist is determining if the algorithm is doing what it's expected to do. They do have a lot of power, they do have a lot of opportunities to agree or disagree with how the algorithm proceeds. But at the same time, the algorithm has been designed to ensure that the variability that we've otherwise seen or the sensitivity that we would like to see for a device like this is enhanced. And that takes away this human variability that we've seen. It enhances the sensitivity and the precision that we want these unique biomarkers to have. And when we submit this, or the expectation that FDA especially has had now, is that when you submit the data, that objective evidence should support the end-to-end workflow. Everything that you did from the start of the time you got your tissue, the way you created your slides, all the way to the point you got your biomarker report, is what we would like to see as an end-to-end system verification and validation data set, and you need to submit the whole of it to us. Any changes in the way the device is currently described would be considered an off-label use if you're using a component in that entire workflow that is not defined in the device description. And that is a very, very important thing to understand, that things like a display monitor. We know that pathologists would use different types of display monitors, but we have to ensure that if they are using the stroke to device for NSCLC with the particular algorithm that Roche is developing, the expectation is that you use it with the monitor with which it was verified and validated. Any changes to any component of the device, you scanning the images or scanning the slides to get that image on a different scanner that is not the one that we NV'd with, would be considered an off-label use. And so the realm of how FDA has always seen device configuration, how they look at objective evidence that supports your intended use, how they look at off-label usage has not changed. But the content of what that device description looks like has moved from us creating one assay kit to probably having 7 to 8 components that are systems of their own. And hence this becomes a system of systems now, which is a very, very high complexity, high risk CDX diagnostic device. So that's where we are.
That is a very big shift in perspective from the test side, but it kind of is in parallel how digital pathology was being viewed by regulators from like the first scanner clearance, right? It was a workflow, and now even the tests are part of the workflow, which I see as something pushing digital in general, digital pathology forward, because now we have something highly complex and highly granular, cannot be reproduced like in a DIY fashion. And this is something that helps patients. So if you want to help patients, you need to expand to be able to provide this kind of workflow. And it's, this is my perception, my like joy, why we discovered this biology in terms of how it's going to go backwards to promote digital pathology, because this is a level of care that we can now provide. Like everybody wants to provide that level of care. And now let's find a way to actually do that. So Gordana, back to you. In this kind of landscape, regulatory, biological, like the limitation of our visual interpretation, when I hear like, okay, it's a ratio of membrane relative to the total stain. Like, even if I wanted, and if I count, like, even if I drew every single cell, I would be tired after 15 minutes of counting these pixels. So that's basically not feasible. So, I want to hear your perspective on why is computational pathology no longer optional for certain companion diagnostics. And here we're talking about the specific companion diagnostics, but I am hoping for more discoveries like that. And also I am hoping for the other estimates that pathologists have to do to no longer be estimates, but have computational support for that.
Thank you, Alex. So as we move toward precision medicine, the shift to quantitative data in precision medicine makes manual biomarker scoring with necessary reproducibility nearly impossible. So the human eye, as remarkable as it is, has physical and cognitive limits for the precise quantification of modern precision demands. So this challenge is leading the field to transition toward computational pathology. And as even Saleh mentioned, instead of tiering scoring system, algorithms can generate a continuous score by counting every single pixel and cell. Basically, a computational pathology produces a continuous scale, it was mentioned earlier, and the key metric is a normalized membrane ratio, which precisely quantifies the cell surface to internal cytoplasmic protein proportion. So when we talk about pathology, in traditional pathology, the instrument is the microscope. But the measurement happens in the pathologist visual cortex. In the algorithm integrated system, every link in the multi-step computational pipeline impacts the final measurement. When a project starts as a manual score, it can fail because the human eye imposes artificial categories and biological gradients. So, for example, pathologists might score a tumor as 1+ or 2+, if the true biological threshold for drug benefit is actually 1.4. The manual score is too noisy to find a correlation. Half the 1+ patients respond and half the 2+ patients don't. So the biomarker looks non-predictive. But measuring protein density with the algorithm on a continuous scale eliminates the subjectivity, this variability, and noise present in human assessment. In this case, patients with a score equal or above 1.4 can show a survival benefit. Further example, the industry saw this play out in real time with TROP2, we already mentioned, in small cell lung carcinoma. And the clinical utility of tropion initially appeared to be inconsistent when assessed with manual IHC scoring. The results didn't clearly show that more TROP2, better response. So the manual score couldn't find the right patients. However, the algorithm could identify spatial patterns and membrane-specific intensities that pathologists couldn't quantify. The drug became accessible, unlocked, and to specific subpopulation that manual scoring failed to identify. So this is leading to, let's say, algorithm-only biomarkers in clinical trials, which have no manual equivalent. So, but I, as a pathologist, could say that, and also want to emphasize that the pathologist is not replaced. And instead of digital tool as a biomarker calculator providing the numerical aspects, while the pathologist focuses on the complex diagnostic integration.
Mm-hmm. And I love that, because burdening the pathologist with these like calculations that we are like human vision is not made for, is basically taking cognitive power from all the other things that a pathologist is trained for and is very good at. And that kind of supports the theme of collaboration, human with machine, or however we want to call it. But basically now, okay, the pathologist is very good at morphology, recognizing which cells, recognizing is it from the correct compartment, is the calculation being done on the correct things that the device is supposed to do calculations. And that's easy. That's like no cognitive burden, no like depletion from your diagnostic superpowers as a pathologist. And then you have this calculator, like you mentioned, that was designed as a calculator, is a good calculator for whatever we designed it to be a calculator for. And following on that one, I want to ask you, Purvi, about what is the role of pathologists in a computational companion diagnostic workflow? Because you were mentioning, okay, before it would be a pathologist under the microscope, and that would be the ground truth, the estimation of whatever the positivity was, and now we have a full workflow, and a pathologist has a role in this workflow. What is this role, and also how is it viewed differently by regulators? And I would also add to this question, by the team that is working on this workflow. It's no longer like a slide stained with IHC and the pathologist is the test. There's a lot more to it. So what would you say the role of the pathologist is in this computational companion diagnostic workflow?
A very important one. So what we know is that there is nothing that we are creating with these tools that takes away the work of a pathologist or the expertise of a pathologist. Like you and Gordana mentioned earlier, this is more like outsourcing that calculator job to a tool that we know is trained and is precise enough to give us a good output that we can trust. These were the questions also highly discussed in our interactions with FDA, and the emphasis on clarifying the role of the pathologist in this clinical workflow was paramount to us explaining what the device was all about. The first step that we would see as how is the pathologist involved and how does the pathologist interact with a device like this is what we call the assessment portion of how the algorithm is performing. So the pathologist is in charge here. The pathologist is controlling what is going to be the outcome here. The pathologist is able to review if or not the scanner that was used to scan the slide and give the image to them on the computer screen was equivalent, if not better, to what they would have otherwise seen on a very, very sophisticated microscope. They would also look at the kind of staining that was done for that image. If they do not agree with the kind of scan or the stain, they are in complete power to reject it and ask for either a restain or a rescan if they don't agree with the kind of quality of image or the kind of quality of staining that they're seeing on the computer screen. More importantly, they also have the ability to judge the sample quality and what the tissue looks like, because what the human eye sees under a microscope, even if this was a 40x zoom versus what you can see on a computer screen at a pixel level, is a whole difference. It's a whole new world of looking at your sample and your tissue. The slightest of the fold, the smallest of the air bubbles, the tiniest of a pen mark, anything that could have smudged your slide from anywhere, all this gets captured and amplified because you're looking at this on a high resolution screen. The pixel pathway from your medical grade scanner to your image management platform has been evaluated, vetted, and tested so many times to ensure that the image quality that you get on your screen is doing justice to what a human eye would want to see and explore further. So the pathologist is in a lot of control when they get to see the image for the first time, the scan, the tissue for the first time, the staining intensity for the first time. Once the algorithm starts doing its job, the pathologist also has the power to reject areas that they do not agree with, which the algorithm might have selected. So they have a lot of control there. And at the end of the workflow, after they see the biomarker status, because they are the doctors, they are the people who know exactly what the patient history is, what the clinical history is, have the ability to still go ahead and reject the outcome if they don't agree. The plus point here is they can reject, but they would be asked to tell why they would be rejecting a case and why they don't agree with the biomarker analysis, to ensure that even the workflow is able to understand and justify why this was not the appropriate diagnosis. Additionally, it is tools like these and having a platform for an image management system that allows you to make your adjudication or getting a second opinion so much easier than what it used to be, where we would have physically transported a slide from lab 1 to lab 2, which was probably miles away. Pathologist looking at it under a different kind of a microscope to get that second opinion is now happening at a push of a button. And so the pathologist is in a lot of control in trying to also get a second opinion and find out if what they're looking at, or if they are not agreeing to the algorithm analysis, where are those differences lying. And when these things were explained to the agency in terms of our end-to-end clinical workflow, they realized that this is not a black box where pathologist does not have the power to control how the diagnosis is coming out. They have a lot of control, they have a lot of power, and at the end of the entire workflow, they still have the option to say no and get a second opinion if they like.
This is, and Gordana, tell me what you think about it briefly as well. But I think what you just said, Purvi, is very empowering to pathologists who sometimes come into this space with a little bit of fear when a new technology that is a technology that provides something that they cannot provide, there is fear, okay, will we even be relevant? But this again highlights it's a tandem work, and the like the good thing, the thing that is empowering is that the pathologists already have the knowledge necessary to be a partner in this workflow. It's not that, oh, now everybody has to get a fellowship in computational pathology. No, the expertise, the training, what pathologists bring into the equation, they already have that as physicians, as pathologists. So the missing piece of the puzzle is, okay, what does this test do, and how do we provide the expertise that we already have? It's gonna require, Gordana, what do you think about that? Do you feel that way when you talk to fellow pathologists who don't know about it? Do you feel like they think it's empowering, or do you still see some hesitation? And we're going to have questions in a Q&A. I'm super curious what the
probably still have some hesitations. But moving forward.
That's why you're here, right? That's why you're trying to explain it, and it will require doing things differently by the pathologists, but also in the labs. So, Saleh, a question to you. What must laboratories consider if computational companion diagnostics become routine? What do they need to now think about that they didn't have to think about before?
Yes. No, thank you for the question. It's very relevant and important question, actually. So as computational CDX becomes more routine, there are a few categories or items that labs need to keep in mind when transition or adapt to more digital workflows. First and foremost is the infrastructure and security. Of course, validated digital scanning and computational infrastructure will become an essential component of these diagnostic standards. So they need to implement rigorous measures to address any cybersecurity, for example, throughout the entire end-to-end system to make sure that data integrity and in general, system integrity, all the patient information, it's all kept intact. The other important aspect is process control. And by that, I mean the pre-analytical staining consistency and all these steps that may not have been as important as before, and we had more wiggle room and more buffer to allow for variability. Now they are significantly more critical than before. And the reason for that is these quantitative algorithms, they are very precise and highly sensitive. And they can measure slight variability in sample preparation or staining protocols. And these variabilities, they directly and perhaps negatively impact the algorithm's performance, its accuracy, and also its reproducibility in generating the same results. The other aspect that I wanted to also touch on is the training and interpretation by pathologists. I think in the previous question, Purvi highlighted the role of the pathologist in the the CDX kind of a device. And as Gordana mentioned, we are not replacing pathologists whatsoever, not at all. We are just empowering them with more quantitative, more precise and reproducible tools. So that will require, in my opinion, a major cognitive shift, both in pathology teams and perhaps even oncologists. So they must move away from relying on visually estimated categories like TPS or 2 plus 3 plus bins to interpreting now precise quantitative data. And this might demand, as you mentioned, it doesn't demand to go back and get a degree in computational pathology now in the universities, but it might demand new skills, such as, like in pre-score digital refinement. Purvi mentioned a few examples, like they need to now ensure the scanning was done properly, the quality of the scan is on par with their expectation, the staining, the sample, it's adequate, that we have enough tumor cells present. And also they might be asked to review the image analysis results to just to QC and say, okay, yes, the algorithm is paying attention to the right part of the tissue, has identified proper enough tumor coverage is there, the purity of the tumor region segmentation, for example, is there, and they approve. And this is something that they didn't have to do it before. But of course, it doesn't need a separate degree, just that shift in their training and their mindset. Also, kind of a post-score interpretation, before they used to look at this categorical, scoring. Now they need to understand how the continuous metrics, such as the normalized membrane ratio that we talked about, looking at all cells, the expression of the protein in membrane versus the entire tumor cell, and then look at the 75th quantile of all the tumor population that exists on the slide. That may not be something very intuitive that they can quickly confirm. Just like before, that they could say, yes, I agree, this is a 1+, or yes, I agree, this is the 2+. This is a new way of scoring the IHC slides, and they just need to, get comfortable with and understand how these new computational CDX compute the final binary therapeutic results.
And I'd like
to
add.
Go ahead.
I'd like to add. This is where the challenge lies for us as manufacturers, right? The entire adoption of creating and having a digital workflow in your lab is not going to come easily unless you make that workflow easy to adapt. If you put too many challenges, if you make it very hard, the hesitance will only increase. The adoption will only get delayed. So it is striking that fine balance where it's not an app on your phone. You can't pick it up and take a picture and analyze tumor cells, but at the same time, it's not even that hard that you cannot, get used to it. And going back to your point, Alex, where we don't want them to go and have a whole new skill that they need to learn. And like Saleh mentioned, have that paradigm shift in the way they have been analyzing or diagnosing patients earlier to moving to a new way of diagnosing patients, but having the same power that they had back then.
I think so. This kind of parallels the push on adoption of digital. So here it's on the training level, on the literacy level. There is going to be a requirement of digital pathology literacy at a specific level. Artificial intelligence literacy, image analysis literacy. But this is no different than it was like IHC literacy in the 70s, molecular pathology literacy, which that's far more complex for me, at least as a veterinary pathologist, because this is not as developed as in human pathology. But basically that's another level of literacy that the pathologists need to acquire. And there are different ways, there are different, like this webinar, right? This is one way of acquiring this level of literacy. But thank you so much. Thank you so much for answering all the questions.